pulse el botón Iniciar emisión del panel de control de GoToWebinar para que los asistentes le oigan. El sistema le avisará cuando comience la emisión. La emisión está comenzando. Todos los asistentes están en modo de solo escucha.
Hello.
Hi.
Hi, Carla.
Hello.
Hi, Gabi. How are
Good
you?
morning.
Hi, Elia.
I'm fine. And you?
Fine, also.
I think we are waiting for you. Okay. Elia, dove stai? A
In Umbria,
Napoli?
Gabri. No, sto in Umbria.
Mm.
Abbiamo una casa qua e siamo venuti l'8 marzo. Così abbiamo un po' di spazio all'aperto per i
Oh,
ragazzi.
si sente abbastanza.
Mi senti male?
C'è un po'
Io c'ho una
di
brutta,
eco, sì,
c'ho una
connessione.
brutta connessione.
Ok. Se... Carla, can we see now the presentation?
Yes, we are seeing your presentation already.
And so these people, okay. Your screen, and can we try yours, Elia? Ah, Paulina has arrived.
Ok, do you want me to share my video?
Just to try.
But you have to make Elia presenter, Gabriela, like the last time.
Gabi?
Yes. Hi, Paul. How are you?
Okay, good.
Okay. We cannot see you though.
Let me check. Turn my webcam. Okay, now.
Okay.
Ciao, Paolina.
Ciao.
Okay,
Yeah.
Gabri, I think you can see my right.
Fine.
Okay. Can you see the mouse?
Yes.
Yes, yes.
Okay. Yes.
Okay, great. Can you hear me? 1, 2, 3, 4. 1, 2, 3, 4.
Yes.
Can you hear
No.
me?
No. It's very. It's fine now.
Okay. Okay.
Okay.
Trying to close all the.
I think we have a few minutes. Thing is that I really don't know what people can see. So, for example, now I am seeing my my screen, my
Yeah, I can see your screen.
Yes.
Okay.
Yeah, and I can see you and Paulina.
It's full screen now. Okay. You can see
Because
me
you
and
are.
Paulina. Yeah. When I see my full, I cannot see you. Okay. Great. And what about my control panel? Can you see that I am moving that one on the monitor or not? The control panel.
No.
Okay.
We can only see your image and your.
Let's see how many we are. Carla, where can I check?
32.
I think it's 32, yeah.
Yes.
Hmm. 36. So maybe we wait another few minutes.
Yeah.
Let's see who is here. Daria, Abraham, Christopher, Marco, Vladimir, Samuel. Okay. What do you think? Would you like that we arrive to 40 and then we can start? 40 attendees?
Yeah.
Okay, we are 40. Okay, so good morning and welcome to the seminar series on extracellular vesicles. Welcome registered attendees and very warm welcome to our speaker today, Dr. Elia Dischiavi. This is our agenda today. So after this short welcome, Paulina Ramos, PhD student in my lab, will introduce the speaker, Elia, and then we will listen to Elia, and after that one we have answer question section.
Okay.
We we have regularly space tweet on Twitter, and this is the tweet of the day. I hope that Elia likes it. So we are quite active in the social channels. So the sponsors of this meeting, of this seminar series, is VES4US, Civica Society, and XMS Lab. So you can meet us or see us here also. So just briefly our program, because you can notice that it's now highlighted by yellow, and this is really the the 10th. Actually, the supposed to be the last seminar, but we had the coronavirus emergency, so we just missed these 3, which are highlighted in red. What we would like postpone this one, and we are organizing suitable dates for these seminars, and we'll let you know on social channels the exact dates. I think with this one, I would like to call Paulina to introduce our speaker.
Okay. Good morning, everybody. Today in our seminar series, I have the pleasure to introduce one of our collaborators here in Naples, Dr. Elia Dischiavi, who is an expert in C.elegans and has been studying them, these little bodies, for more than 16 years now. Dr. Elia obtained his PhD in genetics from the Federico II University in Naples, and since then he has continued working in the C.elegans model at the CNR. And I think from 2010 to date, he's in charge of the C.elegans genetics lab in the Institute of Bioscience and Bioresources in CNR in Naples. And he has developed different protocols to study neurodegenerative diseases, applying the C.elegans model. And that among his research interested, and the one that has led to the collaboration between our labs, is he's interested in develop interdisciplinary and new techniques where the C.elegans model can be applied And in today's talk, he will explain us about the application of C. elegans in the extracellular vesicle field. And welcome, Dr. Elia, and happy to see you again.
Okay, thank you, Paulina, for this very nice introduction. And I just would like to mention that if you would like to ask Dr. Elia Dischiavi any questions, you can type them in the questions tab within your control panel. And I also would like to remind you that this section is being recorded. So with this one, the floor is given to Elia. Okay.
Great. So good morning, everybody. I'm gonna thank the Gabriel, of course, for giving me the opportunity to speak about how C. elegans is helping the community in studying EVs, and in particular what we are doing in my lab. I also want to thank Paulina for the nice introduction. I'm trying to exclude the webcams because I'm not able to see the, yeah, okay, perfect. So great. Hope you can see my screen and my first slide, where again I'm slightly changing the title to make it more clear. But I think the tweet from Gabriel already explained it very well what I was meaning in my title. So as they said, I'm working in enabled SADA BBR, and I have quite an old experience in C. elegans, while more recently I was involved in EVs. So today I will give you more something not working. I will give you
sometimes maybe it's slow only, no?
I don't know. Looks like it's not managing the PowerPoint presentation. Yeah. Okay. Yeah, working. So I will present you the model C. elegans as a model system. Then we will talk about. I will talk about a little bit what has been done in C. elegans field to study EVs, biogenesis, and function. And then I will end with a few slides on what we are doing on using C. elegans to study EVs, which is more recent and unpublished work. So as you can see from this image, C. elegans is almost simply a tube with a mouth and opening in the anterior part, and a vulva, which is another opening at mid-body. It was established not many years ago by Sir Sidney Brenner. Sidney Brenner was already famous for his work on phage and bacteria, and in the beginning of the 60s, he realized that phage and bacteria had some limitation for the question he was trying to address, and he wrote to Max Perutz actually a letter where you can see an excerpt here, saying that classical problems of molecular biology have been solved, and the future was laying in tackling more complex biological problems. And for that, he decided to look for new model system, at the same time simple, that can be manipulated, but also with multicellular organization. And indeed, after 10 years, the first paper on C. elegans used as a model system came out, which is actually the first paper on laying the foundation of C. elegans as an animal model, and is depicted here. Of course, C. elegans as other animal models has advantages and disadvantages. So of course, it's important to have a high number of embryos if you want to use an organism as an animal model. C. elegans has the advantage of being very cheap. It has a very good access to C. elegans. Micro manipulation is limited because of the size of C. elegans. The genetics is very good, and gene inventory is known since 1998. One of the reasons why we have so many embryos is because for sure the life cycle of C. elegans is very short. So compared to Drosophila or mice, where you can see that the life cycle lasts for days in Drosophila, or for even a couple of months for mice, in C. elegans we talk about days. So in 3 days, depending on the temperature, you can have from a fertilized egg, you can have a fertile adult. You can see that in 9 to 12 hours, the embryogenesis is completed. And then we have 4 larval stages between the embryogenesis and an adult is capable of laying eggs. I also want to mention the fact that C. elegans is a self-fertilizing hermaphrodite, meaning that a single animal is able to lay up to 300 eggs, which will be, of course, isogenic clones. Since many of you in the audience, I think, are physicists, and I know that you are interested in which are the dimensions of C. elegans, I have to say that C. elegans is very small. An adult is approximately 1 millimeter long, a diameter of 80 micrometers, and has a weight of approximately 1 microgram. Of course, this is very important in the field of EVs, where we have to distinguish between different size of vesicles. The advantage is that we can have billions of animals on a very small plate, so we don't need large facilities. And in this graph, you can also appreciate how the volume of the animal is growing during the 4 larval stages, and also, of course, the length of the animals grows during the life cycle. An important, a very, very important aspect of C. elegans is the fact that we know almost everything about the anatomy and the development. And most importantly, the cell lineage is completely invariant. So if you look at the division that from a fertilized egg give rise to a embryo that hatch, the number of division, the timing of the division, and the final number of cells is always the same in all animals. So at the end of embryogenesis, we will have always 20 intestinal cells, 20 muscle cells, and so on and so forth, which makes, of course, the analysis very, very easy and standardized. So in this picture with the Davy staining, you can appreciate all the nuclei in adult C. elegans. Of course, there are a mix of germline and somatic nuclei, which somatic nuclei in an adult will be 959 precisely. Most of these cells are neurons. In this scheme, you can see that most of the neurons are located in the head, the so-called brain. Of course, it's not a brain of C. elegans. And there are 302, at least in the hermaphrodite. This is the intestine in green. In blue, you can see the gonads. The uterus is here with the embryo, which will, after 3 hours, be laid and complete the embryogenesis in the environment. Since we know many, many things, we assign, John Sulzenstein and collaborators assigned a name to every cell. So you can probably see that every neuron here has a name, and this is also true in the tail. And these days, the only animal model in which the connectome, so the connection between neurons, is completely reconstructed is only C. elegans. Another aspect is the transparency. So both embryos and adult are completely transparent. You can see inside, and probably you can appreciate in this video the fact that if you have neurons expressing GFP in the head, you can easily look at them, you can easily study them in a living organism, of course, because this animal is moving on the plate. So we can study cell biology live, and we can do that in the intact organism. And this is very important for EV's field. Thanks to the transparency, Martin Chalf in 1994 used it for the first time in a metazoan the GFP. This is the cover of the paper, and you're probably aware that Martin Chalf got a Nobel Prize in 2008 for this discovery. Of course, as I told you, genetics is powerful in C. elegans. We can manipulate the animals. The genes are pretty much similar to a human genome. So we have 20,000 genes in C. elegans, but the genome is very small. So we have only 100 million bases composing the 6 chromosomes in C. elegans. We can microinject every kind of DNA or RNA. We can make transgenic. We can overexpress or silence by RNAi the gene. We can make deletion. We can humanize the animals expressing pathogenic version of the human disease genes. We can ask for a mutant to the stock center. It only costs $10. And of course, we can make genome editing, like using ZFN, talents, and more recently, CRISPR-Cas9. So any kind of manipulation, genetic manipulation, can be done in C. elegans. This is a list of some of the most important discoveries which have been made in C. elegans. I just want to highlight the one that gave the opportunity to researchers to get the Nobel Prize. As I said, in '74, there was the identification of, for the first time, of mutations that affect animal behavior by Sidney Brenner, who got the Nobel Prize in 2002, together with John Stulston and Bob Horvitz, which discovered for the first time complete metazoan cell lineage, and Bob Horvitz discovered the apoptotic genes, which are completely conserved in mammoths. Another important discovery is the use of GFP, as I told you, in 1994, and the elucidation of the RNA interference pathway by Craig Mellon in 1998, which gave them the possibility to win the Nobel Prize in 2006. More recently, C. elegans has been used as a model also for human diseases. You have to bear in mind that more than 70% of C. elegans genes have a significant human match, and that 75% of human disease genes have an ortholog in C. elegans if you use this threshold. So if you look at some of the important disease genes in humans, I don't know if you are aware of presenilin or SCA1 or SMN for spinal muscular atrophy, you can see that there is an ortholog in C. elegans with a certain a certain value for similarity. Even more strikingly, pathway are also conserved. This is an example of the RAS pathway, which I probably you are very well aware of. And you can see that it's completely conserved in C. elegans, and many of the genes of the RAS pathway have been discovered in C. elegans. And in fact, the name resembled the fact that it was discovered in C. elegans. Usually when you see a gene with a name with 3 letters and a number, this is because it was discovered in C. elegans. SOS is an example. Of course, so many disease models were made in C. elegans. You can probably see the fact that there are Alzheimer's disease models in C. elegans, Parkinson, muscular atrophy, cancer, and so forth and so on, for the long list of disease models already obtained in C. elegans, which are under investigation. The fact that the animal is so small allows for screenings, both for drug screenings and for genetic screenings. These screenings can have a low throughput if you screen for a low number of molecules, but still you can automatize this screening using a worm sorter. You can grow animals in multi-well, so you can imagine how many animals you can grow in a single plate, where you can add, for example, drugs. And this was used using readout as a transparency, for example, for lethality or eggs. For enzymatic activity, you can use movement readout or a fluorescent biosensor, and In the past, Pfizer has used this approach, and Evgen, which is now Syngenta, has also used this approach, as stated by this publication and patents. But of course, as technology gets better and enhance the power, we can have a higher throughput. For example, this is a scheme of a medium throughput drug screening, where up to 2,400 chemicals can be screened per week, using a robot to prepare the plates, a sorter to have a single animal in each of the wells, and a machine which is able to automatically make the phenotyping. For example, in this case, this is Opera actually, from Perkin-Elmer, which is a confocal imager for eye content screening. So you can identify hits in your plate which affect a phenotype or a behavior in C.elegans, as in this example in Nation in 2006. And we are using a similar approach in collaboration with Tijem and Diego Medina, and thanks to a grant from Teleton on a model that I will present to you later on. So if you're interested, we can discuss about that. But of course, throughput can get even higher and better. We're using microfluidics. I invite you to look at the video on YouTube about Hotel Nematoda, which is talking about worm hotel, which allowed to study up to more than 5,000 worms in a single experiment. The idea is to make chambers, small chambers, where single worms are able to grow and for the entire life, and then you can follow their phenotype and also the lifespan. The dimension of this square is as 1 euro cent, so you can imagine how many animals you can analyze. And using plate carousel and automatization, you can appreciate that how you can get to these numbers. And this approach has been used both in UK and Switzerland by 2 biotech using C.elegans for screenings. Being an invertebrate, it's important to underline the fact that raises less ethical concerns to the public and less bureaucratic procedure. It's highly supported because it completely fulfills the 3 R's principle about replacement, reduction, and refinement of higher animal use in research. Of course, it's highly supported by European Union and by Italian law. And you are probably aware of the fact that last year, Sanger Institute in UK closed the animal research facility, suggesting that we really need alternative animal models, and C.elegans can be one of those. In fact, it has also been used for toxicological studies, which is something related to what we're going to talk about in a few slides. This is a comparison made by John Friedman about a number of animals you need for a toxicological study in mice and in C.elegans, the time that this experiment need, and also the cost, which is completely uncomparable. And in fact, another important aspect is the fact the correlation between a healthy 50, so the lethality, the concentration causal lethality of the 50% of the animals in C.elegans and the rat LD50 are similar to the relationship between mouse and rat LD50s, and this correlation is stronger than a correlation of the when you use cell culture, suggesting that C.elegans is much better than cell culture for toxicological study, is a valuable model for predicting chemical acute toxicity in mammals, and this is why it has been recently adopted by National Toxicology Program and FDA in US. This is an example related to vesicles. In particular, they are using in this paper nanoparticles, and you can see that the lifespan, so the number of animals that during time is alive, which in wild type is like that, in this case it was 35, is decreased when you use nanoparticles with silver, as you can see with this dose or an higher dose. This is not true with other nanoparticles, but this means that you can make an experiment with hundreds of animals. Just in 1 month, you can discover the toxicity in terms of lifespan. And of course, you can double easily this number to 2 months and still affordable for a research team. This has been also tested for the gold nanoparticle. I'm just giving you a couple of examples to understand how you can use C.elegans for toxicological studies. And gold nanoparticles are useful as carriers and can be used with different dimensions. You can see that the smaller is the gold nanoparticle, the more is the effective on survival and broad size. Higher size of gold nanoparticles have less effect, but this is also it's also possible to, in C.elegans, thanks to transparency, to see where the nanoparticles are entering, where they are distributed. From this picture, it's clear that they are entering from the mouth, so there is no entrance from the cuticle, which is impermeable. And you can see that the localization of the small gold nanoparticle is more clear in the anterior part of the intestine, while the bigger one are more clearly visible in the posterior part of the intestine, possibly also related to different pH of the intestinal lumen. Of course, there are many disadvantages using C.elegans. For example, there are no organs like eyes. There are many factors which are not present, such as immune function. There are also cells which are not present, like oligodendrocyte. There's no myelin, and these, of course, are critical components. There's no inflammation, and it's another example. So, which are critical components in many vertebrate physiological processes. Moreover, even if a gene is conserved, it happens that there is a missing part which does not allow to use C.elegans as a model. And this is the case of APP, which is the gene that causes the formation from which the beta amyloid plaques are formed in Alzheimer's disease. And the gene is conserved in C.elegans, is called APL1, but the composition, the amino acid sequence is different, so there is no beta amyloid formation and no plaques, just to make an example. Okay, so I think the first part is completed. We can go to see what has been done in C.elegans to study EV's biogenesis and function. One of the biggest issues in EV field is about how you can study EV in a living animal. So how you can understand the function and the biogenesis of EVs without looking at the whole organisms. And of course, C.elegans can be used for this to address this question as an invaluable in vivo system to establish how EVs affect development, for example, behavior or disease states in metazone. Moreover, since we have powerful genetic tools, we can then define the molecular mechanisms of EV release or uptake, or whatever is your question about EVs. Recently, there was a paper establishing the protocol to isolate extracellular vesicles in a growing culture of C.elegans. So from a liquid culture, they were able to isolate the EVs secreted from the whole animals. As a questionnaire paper is still on bioRxiv, but there are interesting aspects that can be discussed. This is an overview of the protocol. Actually, they found that EVs are mostly present in these elution volumes. This is the average dimension of the nanovesicle in these elution volumes, which are the protein content is depicted here, and you can see that it remains the same even after using an exosome spin column, so meaning that this is an homogeneous population of, suggesting that this is a homogeneous population of EVs. I'm sorry. And there are also pictures using TEM of the vesicles, which are here. But I think the most interesting thing are a couple, like the fact that the yield and the size is similar to those obtained from human cell culture, and that they made some proteomics and RNA-seq analysis, and they found that 31 of the 85 most common human EV proteins are represented in proteomic results, suggesting for a conservation also in C.elegans in EV production. But most of the work has been done by 3 labs interested in development during embryogenesis, in algae formation during larval development, and in a special behavior of the animals, which is the male tail chasing. And these 3 aspects have been addressed by 3 different labs: Han Femen in Europe, Michel Abouès again in Europe, in France, and Maureen Barr in the US. And I will discuss a little bit about the result obtained in the 3 systems. Let's start from embryogenesis. This is a movie of an embryo starting from fertilization. So this is the male nuclei, this is the female nuclei getting inside. So you will see all the divisions, because also the embryo, I told you, is transparent. So we have many divisions. The size of the cells becomes smaller. You can still see some of the nuclei. And what women during her postdoc found is that if you silence a gene, which is STAT5, you have a disruption of the embryogenesis. So the embryo are not able to do complete the embryogenesis and are not able to hatch, which is instead clear in the mock. So studying this gene, they found out that during the very first division, so the four cell stage, which is something like here, you can see, there was an accumulation of a marker that they used, which is M-chair refused to fragment, which allows the protein to go into the membrane. So the membrane are fluorescent in the control, but become even more fluorescent in the mutant. And they discovered that this is because a massive release of EVs in the mutant between the 2 cells. So this is an enlargement of this part, and you can see that in the extracellular matrix, there's an accumulation of EVs in the mutant, and is also clear by different TEM looking at the surface. You can see an increase in EVs outside of the cell. They were able to measure the diameter of the EVs. They were able to define the fact that they are coming from membrane budding. So TAT5 role is in inhibiting the secretion of ABs. And actually, I think it's important to underline the fact that TAT5 is human ATP9A, so phospholipid transporting ATPase. Anna Wehman established a lab working on these, and there are many interesting papers that I suggest you to look at. But one I want to show you is about a technique which can be useful also for you. So it's clear that you cannot really distinguish if this enhancing inflorescence is intracellular or extracellular. And of course, it's very hard to follow that just using TAM. So they developed a tool to follow extracellular vesicle with a fluorescent labeling, and they use a diagram for this to this aim. So if you tag a protein which you know is normally expressed with a tag that causes the ubiquitination, you are sure that this protein is expressed where you need, and when it should be degraded, it will disappear, suggesting that you can have a cell marked for the time that you are interested in, and then it disappears later on. This means that the protein in the cytosol will not be visible anymore. But of course, if it has been secreted, so it's outside the cell, so it's not cannot be ubiquitinated, it will be still visible because not degraded. And this is the case using this kind of construct. So you can see that the construct has a tag for ubiquitination, and during time it is recognized and degraded, which are the cells in blue. While where it's not degraded, the cells are still fluorescent, and you can see here in the images. So at the beginning, when the protein is still present and not degraded, you can easily distinguish the boundaries, and after a while the cytosol is not fluorescent anymore and only vesicles are still fluorescent. Of course, you expect that in top 5 mutants this become even more clear, and because there is an enhancement in the release of extracellular vesicle, which of course are protected from degradation. This is clear here, where in normal condition nothing is visible. And I hope from this movie you will appreciate even more what I'm saying. This is a control, so you can see that fluorescent becomes dimmer and then completely invisible, while in the top 5 you can see an increase in fluorescence, and you can see that this is maintained even after when the protein should be degraded, as in this case. I think this is a very useful tool for in vivo imaging of EB. Going to the other lab, in Michel Abouès, they are interested in epidermal and hypodermal cells, and they discovered that there's a special structure in the larvae and in the adult, which are the ali. These ridge here, which are similar to the one that you find on wheels, this is an enlargement. This particular structure is called ali, and is caused by the secretion, is formed by the secretion of special morphogens from the underlying sim cell, which is an epidermal cell. In these images, you can distinguish in the sim cell the formation of MVBs, which contain intraluminal vesicles, and the membrane stacks, and you can then see the exosome secreted in the extracellular environment. They used this model to identify genes important for this process, and using RNA interference and 10,000 clone to be silenced, they identified more than 80 proteins which are important in this process. So when these proteins are silenced, there is no secretion of exosomes and no formation of the ali. And among those proteins, 10 are escort proteins. And I want to just underline the fact that in these screens, they identified the RAL1, which is important for exosome formation and is the homologue of Ras-like GTPase in mammals is called RAL-A and RAL-B. And another important gene that discovered is VH5, which is an ATPase. And these genes came to be important for exosome formation also in mammalian system. And the last example I want to show you is about function, which has been associated with EVs. I told you that C.elegans is an hermaphrodite and is self-fertilizing, but there are also males in nature with very low percentage. They are completely different. Of course, they only carry sperm, so male gametes, and they use the tail to find the hermaphrodite. So you can see a male here and the hermaphrodite. Then they have a stereotyped behavior, which is the mating behavior. They always start finding the vulva from the other side. So the vulva, which is the final target, is here. This is the vulva, the vulva is here. They start going back on the other side. They turn and they locate the vulva to insert the sperm into the uterus. And this is a very well studied phenotype, which came out to be also associated with EVs. In Maureen-Barr, they found that particular transgenes in which some proteins are fused to GFP can be observed outside of the animals. These are those small dots, and so they are secreted, and they found out that those neurons, which are ciliated neurons, are able to release these EVs into the environment. This was confirmed using specific antibodies, and they also found mutants in which these EVs are not secreted anymore. In one of those, for example, KLP6, which is a kinesin 3 protein, the EVs accumulate inside where the cilia are at the ciliary base, and they are not secreted outside. What about the behavior? So they found, among many other aspects, that if they had these EVs from a wild type on a plate and they had a male, after a while, this male moving on the EVs is showing a special phenotype, which is called male tail chasing, which will be seen now. you can see that it's touching his head with the tail for a second. And if you count the number of events per essay, you can see that this is it can happen 3 to 1 time for when EVs are from a wild type. This almost never happen when you use buffer, and if you use EVs from a KLP6 mutant, you will see less of this phenotype, suggesting that these EVs can mediate male mating behavior. And with that, I want to finish the general part and start what we are doing. We started only last year working on EVs, so I'm not really an expert, and I apologize for that. We were involved in the VES4US project to use C.elegans for studying EVs. So we are not interested in studying EVs from C.elegans, but we are interested in using C.elegans as a tool. So one of the things that we are in charge to do is to study biodistribution of vesicles from different sources, especially natural sources. To start, we used labeled liposomes, which were injected into the animal, something like you can see in this movie. We injected the labeled liposome made by Katharina Ladsvetter and Svenja Morsbach, which are our collaborators in VES4US in Mainz, in Germany. They labeled the liposome with II, which is a red fluorescent marker. We injected this liposome here in the head, and you can see that some cells along the body uptake these liposome and become fluorescent. And in particular, I want to show you those cells in the head, which are the so-called coelomocytes. They uptake material from the pseudocellum, which is a fluid cavity in the animal, and they work somehow as macrophages. So we were able to see that the injection of liposome is efficient in causing cellular uptake and distribution along the body. Of course, we are also interested in checking if this is possible by soaking. So if we can mix animals in an epidurf with containing vesicle or liposome labeled, we then want to test if by drinking those liposome we can study the distribution into the animal and see where we can find this liposome going, and if this is also transmitted to the progeny. And this is something we will start soon. And of course, we're also interested in testing the toxicity of EVs, labeled or non-labeled EVs from natural biosource. But we are a neurobiology lab. We have projects on neuron development, neuron function, and most of all neuron degeneration, both in Parkinsonism and in SMA. So we want to use our skills, our know-how, our reagents to study if we can protect neurodegeneration from neurodegeneration using EVs. And this is something that we have done in particular on our model of SMA. SMA is spinal muscular atrophy. Is a devastating disease in humans caused by deletion of SMN1 gene. This caused the death of motor neurons in children and their death. In C.elegans, we were able to silence the same ortholog in only 19 motor neurons in the ventral cord, these in blue, which normally express GFP and can be easily visualized. But when they die, because of the silencing of SMN, they are not visible anymore. So please concentrate on the ventral motor neurons, which are schematized here. Of course, this doesn't mean that GFP is not expressed anymore, but there is a real cell death that we demonstrated using different apoptotic marker. And like in this case, you can see that the neuron in the ventral core is dying. It has an apoptotic shape and is expressing an apoptotic marker. So we have two readouts. We can screen for survival motor neurons, which is this one, and we can look for a dying motor neuron, apoptotic motor neurons. And the idea is to find drugs, protective compound, or even EVs, or use EVs as carrier to rescue this phenotype and come back to the wild type situation. We already in the past tested drugs and natural extracts able to rescue the number of visible motor neurons. So in the mock, there is no rescue, 0, negative. We try to have a percentage of rescue going up to the scale, and you can see that a couple of experiments done with tomato extracts from different parts of tomato were positive in rescuing the number of motor neurons. So in collaboration with Gabriella, Oxfalvi, and Ramesh, we decided to test if nanovesicles from tomato were accounting for this kind of rescue. So we used the nanovesicle isolated from Ramesh, and Ramesh a couple of appointments before already talked about that. If those nanovesicles obtained by ultracentrifugation can be neuroprotective by adding them to the plates, adding animals, and after 3 days observing the number of motor neurons which are dying. And indeed, we found out that nanovesicles, so the smaller part, which is different from microvesicle, both because of the dimension and for the preparation technique, and Ramesh already explained to you how to obtain a microvesicle and nanovesicle. You can see that nanovesicle can make a partial rescue of the number of motor neurons which are dying in our model. Of course, in the wild type, there's never a dying motor neurons in the ventral cord, while we are having a partial rescue in nanovesicles. This was true with a blend of tomato. They found also a similar effect. We found also similar effect with different nanovesicles from Piccadilly tomatoes. You can see that nanovesicles have a partially rescue the number of dying motor neurons compared to the mock, to the control, and they do work better compared to microvesicles. Of course, the question here is that we don't want to stop the apoptotic path. We don't want to stop the dying cell, but we want to intervene early in the process. We want to avoid completely the degeneration of neurons. We want to prevent the complete degeneration of neurons. So we went to the other phenotype, the number of surviving motor neurons which express GFP. In the wild type, there are 19. So this is the opposite, an opposite graph. We want to have a higher number of surviving motor neurons. In the mock, this number is very low. We have a slight rescue with the total extra, confirming the fact that also with Piccadilly we have some neuroprotective effect. When we separate surna pernatant, with which is the part not containing nanovesicle, and nanovesicle, you can see that the only rescues obtained with nanovesicle, which are enriched in neuroprotective factor, and in fact the rescue is higher. So we can intervene even before the apoptotic mechanism is started. Of course, there are many open questions which are still to be addressed, both with tomato, nanovesicle, and other natural sources. We still have to test if there's any toxic effect or if there are other beneficial effects of those vesicles. We are interested in understanding which are the elements in the nanovesicle causing the rescue and why they are different from microvesicles. Is there any direct target to neurons? So once they enter, it's clear that they have an effect, but how is this happening? Is because they arrive to the neuron, or is because they are changing the metabolism in the animal, for example, in the intestine, and these affect neurons? And of course, if this is specific for the small model or also for other neurodegenerative models, which is something we want to test. With that, I want to end by acknowledging the people who has done the work that I presented. Ivan Gallotta, which is actually a postdoc in Tübingen, was PhD in my lab, and he established the small model. I'm also grateful to Nadia Mazzarello, who did the experiment on drug and natural extracts. And this is the composition of my lab these days. I want to thank in particular Pamela, who did the work on microvesicle and nanovesicle from tomato obtained by Gabriella, and also Giusy Zampi. She's the lab manager and research assistant, so she's the pillar of the lab, and all the projects have to deal with her contribution. I want to also thank the collaborators, both in Italy and in Europe. This project has been funded by different agencies, and also is of interest of Artera Bioscience, a company in Naples, who we are developing new tools and new extracts. And of course, I want to finish by thanking the VES4US consortium. Beautiful people. We're having a great time. I hope to meet you all soon. And of course, we're also doing, I think it's a great scientific network. There are many experiments we are doing with many of you, which I didn't have time to show, and which are still in progress, and I hope in the next future to show what is ongoing. And with that, I want to finish by showing you a couple of take-home messages. I want to remind you that Bruce Alberts, in his editorial in 2010, suggested that we will find in a surprising position. As incredible as it seems, future research on flies and worms will quite often provide the shortest and most efficient path to curing human disease. And some years before, Friedrich Nietzsche in Das Boot Zarathustra wrote that you have made your way from worm to man, and much within you is still worm. And of course, I want to thank you for your attention.
Thank you, Elia, for this very nice and educative seminar. I had the opportunity to start a collaboration on this nanotome project with you, and I actually appreciated really the power, and I really impressed about the utility of this model system. For both the biological and the toxicological studies of EV. So a special thanks for letting us know what is in the EV field of C.elegans, and also the utility for these measurements. So with this one, I think we would like to check the questions. If there are any questions from the audience, then at the moment, cancer model, I just see. Okay, I make it a little bit bigger because I can't see from my. Thanks, Elia. Do you think that it is possible to use your system as a cancer model, not solid, but in liquid? And this is the question.
Can you repeat the last part? Cancer model?
Not in solid, but in liquid.
Okay, so yeah, of course, we can absolutely use liquid cultures, and this is what we are doing. For example, in this example here, we can grow animals in 50 nano microliters, and in 50 microliters we can have up to 40 animals, which means that in each of these wells you can have 40 animals, and you can grow them up to a couple of weeks. Actually, we are testing a different condition to grow them even more. Usually they prefer to grow on solid phase, but also in liquid it's possible. About the cancer, I'm not an expert. In general, because of the strict cell lineage, invariant cell lineage, C.elegans is not the best model for cancer, but if you look at the genetic pathway, they're all very well conserved, and also the exosome machinery is similar to the one which is in place in cancer exosomes. So depending on the question, that the real biological question that you are interested in, C.elegans could be useful. I hope this answered to your question.
I see this was only this was the only questions so far. So maybe there is one, yeah, more. On my channel. I don't know whether Carla you can read it because I have difficulties to visualize these questions on my monitor. There is a ciao from Rosetta, but maybe there are no.
Let me check it. Okay.
Can you see some other
Yes.
questions to Elia?
Yes, there's another one saying, when you mentioned humanizing C.elegans, can you throw more light on this?
Yeah, no, I said it.
Can you repeat?
You can tell Abby.
Humanizing C.elegans. If you can tell something more about this aspect.
Yeah, a classical aspect of humanization of C. elegans is the expression of the protein which causes a disease in humans. For example, Huntington protein. So, if you're probably aware that Huntington with enhanced number of polyglutamine stretch is a toxic protein which cause neurodegeneration and neuron death in Huntington patients. So there's a very interesting work from Alex Parker some years ago where they used the human protein. They expressed this protein in the wild-type version and in the mutated form in C. elegans, in a special class of neurons, and they found out that this toxic version of the protein, but not the wild type, was able to cause the death of neurons. So they used the human protein and the human version found in patients, and they obtained the neurodegeneration of neurons. So you can study how this protein in C. elegans is causing neurodegeneration. Of course, you can also find neuroprotective agents, and there's a nice paper on Nature Genetics where they identified the resveratrol as an agent protecting neurons in C. elegans from the neurodegeneration caused by human Huntington. This is an example. Another example is that, for example, C. elegans is not able to taste capsaicin. You know that capsaicin is the molecule in chili pepper which cause the fact that they are chili, and you can humans of course can sense the chili pepper, and C. elegans is not able because they have no receptor for capsaicin. But if you had the receptor from humans in C. elegans, then C. elegans becomes able to respond to the presence of capsaicin, and of course they can have a behavior dictated by the addition of the human receptor in their sensory neurons.
Yes, thank you, Elia. There is another question from Aurora Scorlatti. Did you have a chance to check if EVs have an effect on worms progeny?
If EVs have an
effect on worms progeny?
Progeny?
Progeny, yes. Sorry.
Yeah. No, this is something interesting. So the nanovesicle we tested were only tested for one generation. So on the animal that we are then observing, we don't know if there is any effect on the both on the fertility or on the development of the progeny or on the neurodegeneration of the progeny, which could be also interesting to be tested. This is something we want to do both in collaboration with Gabriella and Nanotom project, and in collaboration with all the VES4US project using a different source for natural vesicles. Yeah, but this is an important point.
Thank you. So another question from Giovanni Pecoraro. If these nanovesicles from C. elegans, do they contain nucleic acids to communicate with other cells?
Well, as I said, in both the example that I showed you, which are I'm trying to go to the presentation. So in both the example that I showed you, the one from ciliated neurons and the one found in the extracellular milieu of growing animals, they found RNA and DNA. And I think the most of the information are from this paper on bioRxiv, where they made an RNA sequencing, and the list of sequences attached to the paper. So yes, the answer is yes, they found DNA and RNA.
Thank you. And other one is from Maurizio Renna. Have you tried yet to feed any of your C. elegans toxicity model with exosomes derived from mammalian cells?
No, that's also interesting to be done. We are establishing a collaboration with a Finnish group on a different project, and the idea is to test the exosome from cell cultures and from patients with different disease to test if there's any effect on the viability or on phenotypes, and also to test if the animals are able to distinguish with the smell the presence of exosomes from cell culture or from disease conditions. So this is something we are going to start next with this specific collaboration with this Finnish group.
Thank you. There is Ekene Anakor, who thanks for your presentation. Also Marco Yeran, saying very nice presentation, best regards from Ljubljana. There is Rita Carotta, who asked a curiosity. She wants to know, when the animal grow, how do you distinguish between ages of animals?
Once it is an adult, this is hard to say because the shape is almost similar. Of course, we can see that if the eggs in the uterus, this means that they are in the fertile period, which is 3 days after being an adult. Only for 3 days they still lay eggs. Then there are the shape becomes thick. They move less. There's an increasing accumulation of lipofuscin in the intestine. Of course, what we do is to synchronize animals. So to follow, to be sure that they are being born in the same day, and so we know exactly how old are they. This is the way we usually we use a time scale to assess the age of the animals. But there are phenotypes which change during the lifespan.
Thank you, Elia. And there is a question from Antonella Bongiovanni. And the question is, if it is known how, which pathway the bone cells use to uptake the EVs?
Which cells uptake?
The uptake, the pathway, the bone cells, I mean the C. elegans cells, they use to uptake the EVs.
No, this is less known. So actually, that's an important question. What I found in literature is more about the pathway involved in the release than in the uptake. For sure, there are a lot of genes which are known for the endocytic pathway. There are a lot of genes which are known for other aspects of the for phagocytic process, which possibly are shared, but I couldn't find specifically something on the exosome uptake, both in all the 3 examples that I showed you. So this is something possibly completely new in the field, at least to my knowledge.
I see. There is one question from Mauro Manno. He wants to know if C. elegans can be used to study other EV-like particles, such as viruses, and in particular virus uptake mechanisms.
I think so. This is a time question. For sure, I can tell you that there are viruses specific for C. elegans, and there are a couple of groups which are actively working on that. So there's some literature on the infection by virus from virus to in C. elegans. Of course, these are C. elegans specific viruses. I don't know about virus from other species. For sure, there's a lot of literature on bacteria. So they're using C. elegans to test how bacteria infect, how bacteria, these are also for human pathogens, which are the toxic properties, and now you can also avoid the infectivity of bacteria. I know less about a virus, but this could be something that I don't know. I'm sure about the virus on which are specific for C. elegans.
Okay. I will try to read what Veronica written. She says that, dear Elia, dear friends, I am happy to see you all well. How do you distinguish between exosomes and MVs, microvesicles?
I don't distinguish. So what I'm reporting is everything on literature, where they also in C. elegans they are using dimensions and the source inside the cell to distinguish between microvesicles and exosome. And there's in those lab which I refer to, they are actively working on that. Regarding nanovesicle and microvesicle, this is more related to the is again related to the dimensions and to the way they were prepared. And this is something that Gabriela probably is more expert and can answer to. I don't know if Veronica was referring to the work in elegans or the tomato vesicles.
We know that mainly distinguish them by size and yeah, sometimes maybe protein contents, but there is really not a huge difference between those. So in our lab we try to use extracellular vesicles like term, and it could involve both, so exosomes and MVs. And there is another question from Veronica, and it's provocative. And this is, in your opinion, what is the difference between virus and exosome? I don't know, it's directly to Elia or maybe to all of us in these days. But thank you, Veronica, for the question.
Which is
I don't know whether Elia you wanted to. What is the difference between virus and exosome?
Well,
but Veronica is saying on the graphs you have shown. Just
on the graph.
There is. I will then read what Antonella is saying.
I didn't get the point. So, what about Antonella?
No, I'm just saying that there is an answer from Antonella, but we are waiting for your answer. If you don't have one,
no.
Maybe
I mean, apart from I've only naive things to say. Of course, the pathogenesis and the fact that the origin, the exosomes are not able to replicate inside the cell, and these obvious things. I don't understand what's the point that Veronica wants to raise.
Maybe because of this recent virus epidemic, no? So
yeah,
just
I mean, the work, it's clear that the work in EVs
discussion going on. Yeah, actually, Antonella answered the same, that there is actually no much difference, but EVs are surely not replicate, or she's sure that they do not replicate, and in fact it has been not seen so far that they could replicate. So yeah. So
I think that it's important to know very well how exosomes are born and what is the function, because many information can be used for probably for other fields, and in particular for viruses. But the way exosome and virus enter into the cells is completely different. So I don't, at least for COVID, for SARS-CoV-2. So there are a lot of differences.
Yeah, well, there are also some similarities, which is pointed out by Veronica, like biogenesis, she says, as they are quite similar. I don't know what she meant on the biogenesis. I mean, maybe the MVs and the exosomes. Maybe she went back to that point, of course. Well, it was a nice discussion. I think also, Eli, I hope you enjoyed that. There were so many questions this time, and
thank you very much.
I think we are to conclude this. We conclude also these 10 seminars. Let's say that temporarily. And I will be back to you as soon as we decided about the time of the and the modality. I mean, how we are going seminars which were not scheduled. So thank you very much, and have a nice day. Take care and be safe. So bye bye.
Thank you. Bye.
Bye bye. Thank you, Elia. Thank you indeed.
Bye.
Ciao Carla. Grazie. Thank you.
Ciao Carla.
Ciao everybody.
How are you, Carla?
Fine. Fine. Staying at home as well.
Great.
See you all around. Have a nice one.
Yep. Hopefully next.
Yes. Bye.
Carla, may I ask? Bye bye. May I ask you if someone wants to, I mean, the 3 seminarists who were not able to present their seminar, if they wanted to do so, can we still use the
platform?
Yeah,
I mean, sure. Yeah. No problem.
Okay. Thank you very much. Thank you very much.
You tell me. You tell me when, and I'll program it. Okay.
What?
You tell me whenever you want to do it, and I'll program it.
Okay. Let's say if they want, then wish to make that, let's say 1 week before, I will write you an email and we can organize. We will try to organize that. Okay.
Sounds good to me.
Cheers. Bye-bye.
Bye.